Good afternoon, viewers at home. I remain Adeoti Olatunde Michael. This evening, we are to look at continue our educational series lectures on advanced toxicology. Advanced toxicology. The In the last video, we look at dose response relationship. We look at dose response relationship. And today, we are going to look at the mechanisms of toxicity, the mechanism of toxicity. So, under this, we are still going to examine cellular and molecular toxicity. That is a mechanism of cellular and molecular toxicity. We will look at the details about enzyme inhibition and activation. What are the mechanisms by which toxic agents will actually block or impact with enzyme activities of the cell. Then we will look at reactive oxygen species, ROS, and their free radicals. How did they look like? Then we will also go on to examine apoptosis and necrosis. And okay, so let's try and highlight what we are going to do today. So we are going to look at cellular and molecular mechanism. Cellular and molecular mechanism. Then we will also examine enzyme inhibition. Okay, and activation. We want to look at that as well under mechanism of toxicity. After that, we will move forward and look at what is called reactive oxygen species, which is called abbreviated as ROS, and their free radicals. After that, we will look at apoptosis, which is a cell death, and how toxicants could lead to apoptosis, that is self-destruction or self-death, and necrosis. So then we will cap it up with what are the toxic effects on DNA and protein synthesis. So at that point, we will just be rounding it up for today. Now, just like it is, I told you yesterday that toxicity, the mechanism of toxicity is very, very central. It is very, very central. Understanding this topic is very central because it is of any nature is able or are able to interact or impact on the biological structures. So we want to actually do this, review this topic so that we'll be able to know how each of these toxic substances, how are they able to have direct effects on the biological system. Don't forget that I told you in the earlier video that 93 that human is exposed to nothing less than 33% and 93% toxicant, not less than that. Okay, and that is why I want to actually represent this with okay, so we want to look at how do they actually impact on human system. So the first one that we are going to consider is the toxicants that we have effect on cellular and molecular mechanism. These are okay. What should we should take note about this one is that number one, this affects those ones that interact. These substances, they interact with the integrity. They interact with the integrity of the cellular constituents, cellular components. Okay, apart from cellular components, maybe cellular activities, such as cell division, cell growth, normal cell growth, respiration, and so on. There are some of these chemicals, once it is being taken in any dosage, in the dosage that is going to that is above the threshold dose, at the end of the day is going to go ahead and interact with the integrity cellular components or activities of the cell division, which also include the cellular components like cellular respiration, protein synthesis, and so on. Okay, so and what really happened is that example of chemicals in these categories are e.g. those ones that have as a surfactants, that is they will interact with, they are always in liquid form. Detergents is an example of chemicals that can cause this. Then we also have those ones that also interact with signal transduction. I told you that signal transduction is a process by which the cell communicates with each other. So for example, let's say you have cell A and this is another cell B. Under normal circumstance, signal transduction is whereby this cell A and B will crosstalk with each other, and by the time they crosstalk, that is when, don't forget that the cell must communicate to know the next process. Before cell will decide to go into cell division, it must communicate. The cell must know that it is the right time to actually go on cell division. So if normally there is a normal way of this one cross-talking through signal transduction with each other. Now when you now have all these surfactants, detergents, by the time this one now exists, if this one has now imparted directly on how cell A and B communicate. That is we're going to fall under cellular inhibition or cellular toxicity, because it has affected the integrity of the cell. Then the cell division will no longer take place normally. Cell will not grow at the end of the day, or even if it grow, some of them can actually lead to overproduction. It can lead to atrophy, a condition that is called atrophy. That is when there is overgrowth, it can lead to cancer. So that is the reason why part of those one that we toxic substances that we impact on the cellular component is surfactants and the other one. Then again, apart from that, apart from having interaction with the cellular components, signal transduction and so, any compound also has its effect on the cell membrane. Anyone that also have its effect on the cell membrane will also be categorized as a toxic substance that can be, that the mechanism of action is cellular. So that is that. Then apart from that, immediately it has interacted with the cell membrane, what is going to do? That means it is going to disrupt the normal functionality of the cell membrane. And when the cell membrane is already being disrupted, you will know that the cell must have lost the ability to communicate. Just like I said, it's going to lose ability to communicate. Cell division will be badly affected, and so on. Now, example of chemicals that do this include mercury, all these heavy metals, mercury, cadmium, because they interact directly with they interact directly with the cell membrane. So, and the third mechanism is disruption. Any substance that leads to the disruption of cellular signaling. Cellular signaling is also categorized in this form, although I've actually said that. And those ones that do that, they have their function by interacting with what is called the G protein. And you know what G protein does? G protein are example. They are proteins that are very special in signal transduction. Okay. Now that is number 3. So the next one, another way whereby which toxic substances can interact or impact on the cellular component is if number 4, that is if they are able to interact with if they interact with directly with mitochondria, mitochondrial protein. Don't forget that mitochondria also has its own DNA, because it is a semi-organelle, it is a semi-autonomous organelle. That is, they have their own DNA. That means they can also carry out their own protein, that's from DNA down to RNA. Then thing that now disrupts the integrity of the protein in this mitochondria is also going to be classified under cellular and molecular mechanism. Or don't forget how important mitochondria is. Mitochondria is regarded as the energy store. So it is the powerhouse of the cell, because that is where you see ATP being produced. So any impact on the mitochondrial protein is going to affect the energy store, and once the energy store is being affected, you know exactly what is going to be the implication of that. Okay, so that is that about whereby all this. So at the end of the day, before I leave cellular and molecular mechanism, at the end of the day, once the cell membrane, so molecules or substances in this category can initiate, they can initiate apoptosis. By the time we get to apoptosis, we will actually consider it, but there are some of chemicals that they have the ability to make cell to be able to undergo the process of suicide. Okay, then again, before I leave that molecular mechanism, anyone, any substance that has to interact with DNA or nucleic acid directly will also be categorized under cellular and molecular um, toxicants. Cellular and molecular toxicants. Okay, so let's look at the other one, which is enzyme inhibition and activation. Enzyme inhibition and activation. Okay, so substances in this category, which are toxicants in this category can exert their effects directly on the enzyme system. It can prematurely activate some enzyme to begin to manufacture and to begin to malfunction. So initially, or let me say prematurely, this kind of toxicants will just go and activate. For example, don't forget the process of gene expression. Under normal circumstance, there is a time that no matter how functional any gene could be in the body, there is a time for the cell to actually undergo and get to the process of cellular rest, whereby the cell will go and rest. You know, after being, you must have overworked yourself. Under normal circumstance, every cell must also go. So, and that's what is called interface. That's what is called interface under cell division. So these toxicants, they now have the ability to go and interact directly. They will go and interact directly with the enzymes that are supposed to, for example, transcriptional factor or RNA polymerase. These two enzymes are very key in gene expression. So if you now have toxicants that will now have their effect on any of these, so that enzyme, the functionality or activation of enzyme is going to be done prematurely. So, and examples of toxicants in this category: organophosphorus, okay, as well as chlorine, then CCl4, that is a chloride. Okay, so these are part of what happened. Then some activation, for example, we know how very important P45 450 is in liver. So there is nothing that will take place, no activity or process that will take place in the liver if there is problem with this enzyme, which is called P450. You know, this one is central because this is where those metabolic product, the function of one of major function of the liver is ability to detoxify. So ability to detoxify, and its ability to detoxify is directly dependent on the functionality of P450. So if this P450 is already being overwhelmed or is already being impacted, that means most of the chemicals that is supposed to be detoxified is not going to do that again, and you know there's going to be accumulation of all these toxicants, and eventually it's going to have its effect on the whole cell. Don't forget that what we are trying to do is we are trying to look at what could be the effect, direct effect of some of these toxicants on biological system. So that is how enzyme uh, are being, can be inhibited, and which will also lead to activation. And don't forget, overactivation of this as well. Overactivation of it means that even the non-toxic substances is going to be detoxified, is going to have its effect on. Don't forget, liver contains the largest amount of enzymes, hydrochloric enzyme, proteolytic enzymes. You have a lot of them, proteolytic enzymes, you have it in the liver. So if this one is now hyperactivated, so that means even water that's supposed to be retained by the body is going to be what? Is going to be detoxified. It's going to be termed as being toxic, and that is a point whereby the liver continue. So some of the processes at which this can be affected include processes of homeostatic condition. You know what homeostatic condition is? Is that ability to bring back the body temperature into, or to regulate the body temperature after being active. That is that. Okay. Now, apart from homeostatic condition, you know that in the liver, especially when we have that is where we have the highest concentration of smooth endoplasmic reticulum, SER. So, and you know the function of smooth endoplasmic reticulum is production or synthesis of fats or lipids. So organophosphorus and some of the toxicants in these categories can also have its effect on the smooth endoplasmic reticulum, and that is telling us that there is not going to be what? There is going to be high deposits of fatty acid or lipid on that membrane. So that is going to make it, this one is going to insulate, and you know the implication of insulation in human, over insulation, that is going to lead to antipyretic condition, or it's going to lead to that. Okay, so let's now consider the third one, which is the mechanism by which toxicants will carry out its function on the reactive oxygen species and the free radicals of the human system or of the system. Okay, so that has the ability to release oxygen, is going to be classified under this, because we know the implication of incomplete combustion even in car engine. How much more is there going to be premature release, or let me say excessive release of reactive oxygen? So whenever the cell is ROS into the system, that means what is going to happen is that most of the excess oxygen will be converted into toxic substances like peroxide, like hydrogen peroxide, and you know the implication of that in the system. So these are free radicals. You know, when we talk of free radicals, where, for example, OH, hydroxyl ion, free radicals like ozone. Apart from that, and part of example of the toxicants that can perform these functions, we have most of our drugs do this. Apart from that, radiation and air pollutants, common air pollutants, they have the ability to react with the oxygen content of the body, thereby releasing free chemicals and ROS, which are going to be toxic to human health. And this is going to, the effect of this is that it's going to lead to oxidative stress. ROS will cause what is called oxidative stress. So, and what do we mean by oxidative stress? That means there is going to be inflammation. There is going to be inflammation, and you know what inflammation does. Inflammatory response means swelling. Some of the body will swell up, and thereby is going to trigger premature release of antibody or of cellular response to this. Then oxidative stress is number one. Number two, it is going to lead to peroxidation, lipid peroxidation. That is a condition. Lipid will become peroxidized, and you know the effect of hydrogen, and peroxide in the system. Then number three, the effect is going to lead to DNA damage, and any effect on DNA, that means it has the ability for the DNA to initiate the process which is called mutation, and mutation is the primary stage of a cancer. You know that. So that is that. Then apart from that, it is going to lead to what is called antioxidant defense. Antioxidant defense. So what are these antioxidant defense? That means there's going to be excessive release or excessive activities of vitamins, especially vitamin E and vitamin C. So that is what is going to happen. So then apart from that, there are some diseases associated with ROS and the release of free radicals in the cell, and these include cancer, cardiovascular diseases like lipid mutation. DNA, I told you that is going to initiate mutation, and mutation is the primary stage or the elementary stage of what? Of cancer. So it's going to have its effects on that. So, and sometimes that is exactly what also happens in Parkinson's. That is a release of antioxidants into the cell. That is exactly what happens. So let's go to the fourth, which is apoptosis and necrosis. So sometimes I told you that apoptosis is these two. Apoptosis and necrosis is actually what is called two types of death that we have in cell. Two types, and different effects can actually lead to anyone. So this apoptosis is suicide, that some cell will now begin to commit suicide, and necrosis, that is natural death of the cell, that is necrosis. So there are some chemicals initiate this process. When death suppose not to come, they are the one that will trigger death. It's just like red blood cell, whereby their lifespan is 120, and in this sickle celler, their own, instead of 120, will die at every 90 days. So you know the implication of that. How much more when you have a toxicant that can trigger apoptosis and necrosis. Okay, so whenever, and there are two certain conditions. There are conditions that could lead to apoptosis, different conditions that could lead to apoptosis, and another one that could also lead to a process that is called necrosis. So this necrosis is whenever cell sustain serious injury, whenever our cell has sustained serious injury, that is when death will come. So it is an irreversible condition, whereby as a result of injury sustained by toxicants, so it can actually lead into that. So, and apart from that, this one is suicide. This is self-destruction. Nothing is actually leading this to to die, but this one is as a result of severe injury that this one has sustained, and that one will now tell us that it is better to die than to be put to shame. So apoptosis is suicide, and it is also called programmed death. Okay, so every cell has that ability to undergo apoptosis. So whenever you see a cell, it's what is going to have been embedded in it. So, but if that is now being triggered by toxicants, so that is what is going to happen. And let's look at what are these chemicals or toxicants that can initiate apoptosis and necrosis. Example is what is called FAS-L and FAS. Okay, that is these two are enzymes that are found in mitochondria. These two are enzymes that are found in mitochondria that they normally initiate the process of mitochondrial damage. They initiate mitochondrial damage and activation of caspases. So that is when mitochondrial cell is being damaged, some other cells will also be dismantled. And apart from chemicals that cause cell death and apoptosis, it is also very possible that in case of this disease condition, stroke, this is exactly what happened in stroke. So you just discover that part of the cell prematurely is dead, and the host is unable to still work again with that part of the body. So there are some of these that will eventually lead to apoptosis and necrosis. And once this one is done, that means that patient or that person has ability, is vulnerable to develop what is called a stroke condition, okay, or causing unnecessary inflammation, causing unnecessary inflammation in the system. So let's go to the last aspect, which has to do with the effects of toxicants on DNA and protein synthesis. On DNA and protein synthesis. And don't forget that anything that has to do with DNA, that means it can have effects on successive generation. Not only in the parents, it can have effect on successive um, generations. So now let us imagine, let us just assume this is a DNA. This is a DNA. Okay. Oh, now let's say that this is G, this is going to be C. Let's say this is A, and this is supposed to be T. Now, chemicals that fall into these categories, they have the ability to come and intercalate, to come and interact directly with the nucleotide bases, thereby causing effect or having effect directly on DNA. And anything that has effect on DNA, once there is a, is okay, DNA to RNA, then to protein synthesis, that is enzyme production, protein to protein. Now, if there is problem at the DNA level, that means that problem will continue at up to the level of a protein. So when you have bad DNA, you are going to have bad protein as well. So that is some chemicals, toxicants, they have that ability to initiate a direct damage on DNA, thereby leading to errors in enzyme production or in protein synthesis. So whatever have effect on DNA is also going to have effect on protein synthesis. So let's look at some of these chemicals. Some of these chemicals that have that effect. For example, nicotine. Okay, radiation can initiate this. Radiation and heavy metals can actually have their effect that will be directly imparted on DNA. So, and thereby, once this one is being impacted, is going to sometimes going to lead to growth impairment. The growth will be impaired, going to lead to growth impairment. It can also lead to premature death of some vital organs. It can also lead to production of malfunctioning or malfunctioned proteins. Okay, then it can also lead to inactivation of ribosomes, and you know how important ribosomes are in protein synthesis. So then the fourth examples of some initiate this one is we have plant um okay, like plant toxins, toxins like pollen can also initiate any of this. Then it can also lead to aging. You see some cells, when they will become so old when they have not actually supposed to get old. And you will not you should know that there's going to be difference between cell that is already getting old and cell that is still at the prime age. So that is a part of what we want to do today on the mechanism of toxicity. Don't forget that why are we so interested in knowing the mechanism by which all these toxicants exert their on biological system. Now, whatever we are going to classify them based on whether they are toxic or not will actually depend on a lot of factors which we have dealt with at the introductory toxicology. Thereby we mentioned those is very very important: route of exposure, very very important, dose response relationship is very very important, and all this. That's where we started to look at cellular and molecular mechanism, and we listed that any toxicant that has direct effect on initiation or affecting the integrity of the cell is going to be classified under that. And I actually give you example of this. Apart from that, we also look at the enzyme inhibition. We stated some of the examples of chemicals, toxicants that are in this category, where we mention organochemicals like organophosphorus, neurotransmitters, and P4, 450, and so on. Then we also look at ROS, that is reactive oxygen species, and what we can call the release of poisonous free radicals. Okay, we look at that. We look at their effects on causing oxidative stress, lipid peroxidation, DNA damage, and antioxidant defense. So before we now consider some of these toxicants can initiate apoptosis as well as necrosis, and I gave you part of some of the effects of necrosis and apoptosis and the condition that could actually help us, can actually lead to apoptosis. Okay, then